All right, welcome to lecture number 2. This is the digestion and absorption lecture. This would be chapter 2, or at least the first part of chapter 2 in the Fink textbook. It's a pretty massive lecture, lots of material to cover. I've split the recording into two because it is so long and to give you guys a little bit more of a natural break versus forcing you to take a break. So let's get into it. Some of the outcomes of this discussion include what's on the screen here. I'm not going to read it all to you, but we're going to start from the beginning and finish at the end of not only digestion, but what happens to the food once it's digested and absorbed, and really follow that pathway of, where is the food that you're eating going to and what happens to it after that. So, good questions to begin discussing before we get into this lecture. What's digestion? And we'll definitely define that. What's absorption? They are very different. Quite often they're used interchangeably, and they shouldn't be. Digestion is the breakdown of larger food particles into their most basic components. For example, breaking a protein down into an amino acid. Absorption is the process of those molecules, amino acid, getting absorbed or being transported or into the body. And so from the digestive tract, which being in the digestive tract is not inside the body yet. We'll talk about that later. When does digestion begin? At what point does it begin? It's always a good question. I do talk about that face-to-face classes quite a bit. Depends. If you're the food, digestion begins once you start to get manipulated. So if you take an apple off a tree, digestion of that apple hasn't begun. If you mash that apple up into applesauce, that is a digestive process that's happened to the apple, not in you physiologically, right? But if we look at the human, at what point does digestion begin? Well, not actually when we put the food in our mouth, but when we start to think about food. And so those processes, right? You start to think about eating an apple, you might start to salivate, some of your other digestive enzymes and juices will start to be secreted in anticipation of eating. So digestion can begin in multiple places. It really depends on what perspective you're taking. And the importance of understanding the process of digestion, absorption, and then ultimately utilization of energy. Why is it important to us? That's another 20-minute discussion that we could have. Ultimately, when we're speaking about food nutrition, then especially in the context of a sport nutrition class, we need to then better understand where is the energy coming from to provide us what we need to perform a particular sport or activity, where are nutrients or how are nutrients contributing to adaptations to what we do in a sporting context, and then even in a health context, how does digestion and absorption affect our health overall? And when digestion and absorption are unhealthy or not working properly, what are the consequences of that? And we'll touch on all those things in this discussion. So, all right, we'll talk or start with some big picture stuff, right? And the enteric nervous system is where the digestive system resides, right? If we look at the organization of our body's sort of control mechanisms, right, of our nervous system. The enteric nervous system is a main division of the nervous system. It is considered separate from the autonomic nervous system, and quite often it's referred to or labeled as the second brain. So our enteric nervous system, which is our digestive tract, it has its own reflexes, right? And people can be, and I'm just going to be very, the term brain dead, right? And so people whose central nervous system has been affected dramatically, right? Their digestion will still work. How do these people still eat, digest, and process food? The enteric nervous system does its own thing. It does not need influence from the central nervous system to function properly. That's not to say it doesn't work in conjunction with the central nervous system. It absolutely does. However, in case of some sort of traumatic experience or event and the central nervous system's influence from the enteric nervous system is severed, it'll still work. Okay, so hunger. We have to start this discussion off with hunger. What causes us to eat? What drives us to eat? It's getting hungry, right? If you were never hungry, you'd probably never eat. Hunger helps us enjoy food in certain ways, right? The hungrier we are, the better food seems to taste, right? So let's talk a little bit about hunger. Firstly, the hunger center of our brain is in the hypothalamus. Hypothalamus receives inputs from multiple places within our body. One of the primary ones is our blood sugar status, or our blood glucose status. And so when blood sugar starts to drop, when blood glucose, more specifically, drops below a certain threshold, and it's slightly different for everybody, hypothalamus picks that up, and the brain loves glucose, loves to use glucose as an energy source. So when blood glucose drops, brain's starting to think, okay, energy levels are dropping because I'm not getting as much glucose as I'd like to see. Therefore, let's find some food. And so hunger gets activated through that process. The process then is ultimately carried out by multiple sort of substances. Hormones are the primary ones. And so ghrelin and leptin, which are two hormones that we'll talk about quite a bit, they are on and off hormones, so to speak. When ghrelin is present, this is secreted by the stomach, it turns hunger on. Ghrelin is stimulated or released by low blood glucose levels. There's also stretch receptors in the stomach that will activate the release of ghrelin. So when the stomach shrinks in size, when there's not enough substance in it, hunger gets kicked in. Leptin is a little bit more complex. We'll get into that quite a bit as well later on, but it has sort of an inverse relationship. The more leptin that is present, and leptin is secreted by body fat, the more leptin that is present, the less hungry we tend to be. So the sight, smell, or thought of food will also trigger our stomach to release hydrochloric acid, right? You can't walk by a bakery. Hopefully you can't walk by a bakery without smelling the baking of bread and cookies and all that, without all of a sudden going, oh wow, all of a sudden I feel hungry now. Well, that's that process of the hypothalamus being activated through those senses, through smell. Once we start that process, once hunger kicks in, then we can also technically say that, yeah, those enzymes and hormones and other gastric juices that start to get secreted, that is the process of digestion beginning. So hunger will end when all those things in the bullets above are alleviated. In other words, when blood glucose goes back up to normal, yeah, hunger goes away. That kind of thing. All right, so what happens to the food we eat? This is sort of the crux of our discussion. So digestion is that term that talks about or that describes breaking food into its most basic components. Digestion can be either physical or chemical in nature. And so the physical act of digestion has two terms that describe it: mastication, which is chewing food, and churning, which is just the process of our digestive organs, especially the stomach, of sort of pushing food around, or what was once food, pushing it around and exposing it to multiple enzymes and other gastric juices. The chemical process is mediated or carried out by something called an enzyme, and you're probably familiar with what enzymes do. They speed up chemical reactions without affecting the reaction itself. In the context of digestion, enzymes will break bonds between molecules to take a larger molecule and break it into two smaller ones typically, or something along those lines. So following the process of digestion, we have the process of absorption, and that is something we'll get into later. Once molecules are absorbed, they are transported and ultimately assimilated or used for energy production through metabolism. And again, these are all processes that we will tap into in this discussion or later discussions in this class. All right, the organs that we're going to cover, nothing too surprising here. We're going to start from the beginning and finish at the end. So we're going to go from the mouth down to the rectum. Those three organs down, at those three bullets down at the bottom of this slide, the liver, the gallbladder, and pancreas, those are considered accessory organs. The reason that they're considered accessory organs in the digestive process is that none of them ever come in contact with the food we eat. The 6 organs up above absolutely do. Those 3 at the bottom, they play a critical role in digestion. Actually, digestion would be really, really compromised if we didn't have any of those 3, but they never come in contact with food. Sort of an anatomical slide here that describes the organs of our digestive system when we talk about them being the lumen. And so we have multiple layers of the organs of our digestive system. So I just want to lay those out for you. You've probably been exposed to them in your anatomy class or maybe even your physiology class, but just to remind you, right? And this is a graphic of your small intestine. And if we look at the layers of, in this case, the small intestine, again, it's pretty consistent throughout the digestive tract. The I would say the integrity or the thickness or the contribution of the layers differs depending on what organ you're in comparison to one another, but the layers are consistent in terms of where they are and what they do. So we can start on the outer layer, and we call this the serosa. This is connective tissue. It provides form. It gives that organ or that tissue that form that it needs. The layer just below it is called the muscularis externa. And so, as you can imagine, this is the muscular layer of the organ. And so, muscles of the digestive tract are striated. And they have a directionality. And so, depending on how they're oriented, when they contract, they can squeeze or they can sort of push foods in different directions. And so, we'll have graphics later that show that in a little bit more detail. Underneath the muscular layer, we have what is called the submucosa. And this is a little bit more connective tissue. We have blood vessels and also lymphatic vessels, which we'll talk about also later, and how the lymph system plays a role in digestion. We have nerves, and we also have lymph tissue in this submucosal level. And then on the most inner layer, we have what's called the mucosa. And so this is the innermost mucous membrane. It secretes, in the case of the small intestine, some digestive enzymes and other things that help protect the inner lining of the small intestine. And so that is the innermost laying. So those are the 4 layers of the lumen. One of my favorite graphics, I think this is from an older edition. This is from an older edition of your textbook. I think they've changed slightly in this latest one, which is unfortunate. They have a similar graphic in this latest edition of Fink. This is the Fink textbook graphic. But a nice sort of demonstration with the digestive tract sort of stretched out. But on the text information, we have the organ, its secretions, and the functions of each individual organ. This is a great study slide, in my opinion. I'm not going to go over it now. We're going to go over it in more detail with the next several slides. So let's start with the mouth. So when we talk about digestion, we got to start from the beginning. Like I said before, we've talked about hunger and all those other processes, but finally, when food gets into our mouth, we can really start talking about the digestive process beginning in all seriousness, right? And so a couple of things that go on in the mouth. First, mastication. That's the physical breakdown of food, chewing, right? And then there's a couple of secretions that occur in the mouth that aid in the digestive process. First, we have saliva. And you're all familiar with saliva, and if I were to ask you what saliva does, hopefully most of you will at the very least say, yeah, it helps moisten our food. It helps us so we can, the food stays wet so we can actually swallow it. Couple other things going on in saliva that are important. First, there's an enzyme that exists within saliva. It's called amylase. Amylase is a digestive enzyme that breaks down carbohydrates. So this is sort of FYI, big picture kind of stuff. But yeah, the amylases are a group of enzymes that will break down larger carbohydrate molecules into smaller ones. So there's something called salivary amylase that exists in the saliva, helps break down carbohydrates. One of my favorite things to have students do, I'll bring a box of crackers to class. We're not in class right now. Go get a cracker. Or go get a piece of dry bread. Start chewing on that dry bread. Notice in the beginning how unsweet it is. Hopefully it's not sweetened bread. And the longer you keep it in your mouth, so keep it in your mouth for much longer than you normally would, so not your typical 20 chews or whatever it should be. And as it stays in your mouth longer and longer, it starts to taste sweeter and sweeter. And after a couple of minutes, if you can keep it in there that long, and that's the challenge, the bread will taste quite sweet, almost like candy. And the reason is because the amylase is breaking down the large carbohydrate molecules into smaller and smaller and smaller carbohydrate molecules. And the smaller the carbohydrate molecule, or the simpler it becomes, the sweeter it tastes. And so we have these starch molecules, carbohydrates, huge molecules, not sweet at all. The longer amylase breaks it down into smaller and smaller ones, the sweeter it tastes on our tongue. There's also mucus, which we mentioned before, to help lubricate food for easier swallowing. And then lastly, in the saliva, there's something called lysozyme. And lysozyme is one of our first barriers, sort of chemical barriers or antibacterial barriers to keeping us safe. And so the lysozyme is an antibacterial that kills bacteria on the food, potential bacteria that might make us ill if it were to get into our body. So 3 big pieces or important roles, I should say, of the saliva. Also, there's something secreted from the tongue. The tongue obviously has taste receptors. The taste of food, another defense mechanism, right? If food doesn't taste right, we're likely to spit it out, right? If food gets rotten or spoiled, it's an unpleasant taste to us, and we will spit it out, right? Some animals like spoiled food and do quite well with it. They don't spit it out, but we have that mechanism. So the tongue's taste receptors not only provide us a joy and let us know what food is good tasting and good for us, hopefully, but also protect us from eating food that's been spoiled and might make us sick. Additionally, from the tongue, there is a secretion of an enzyme called lingual lipase. Lipases are enzymes that break down lipids, which are fats. And so we have these lipases, again, throughout the digestive tract. They will break down the fats. And so this enzyme released by the tongue will break down fats into smaller fat molecules. So that's the mouth. How many times should we chew? What's the ultimate number? The perfect number? There is no perfect number, of course. Depends on what you eat. Depends on how hard you chew each bite. But research says, somewhere between 20 25 chews per bite should be average. Obviously, if you're eating oatmeal, you don't need to chew oatmeal 20 times. If you're having a piece of steak, you might have to go 40 or 50 times. But generally speaking, 20 chews, 20, 25. Once we've chewed our food enough, food will travel down the esophagus. And so the esophagus is literally a long tube that connects the mouth to the stomach. There's an epiglottis in there that blocks off the trachea so we don't choke on food. Food going down the trachea will cause us to choke and possibly die. So the process of peristalsis is a descriptive term that explains or describes how the organs of our digestive tract, the muscles of our organs of our digestive tract contract in coordination to push food or push the bolus of product in a certain direction. And so when we try and eat something upside down, and you can try this too, although be careful, but invert yourself, right? You yogis in the class, right? Do an inverted yoga pose, but then chew on a cracker or something, right? Can you swallow a cracker upside down? Yeah, you can. Can you drink water upside down? Yeah, you can. Why is that? Peristalsis, a coordinated contraction that will help push food down along the esophagus. Generally speaking, we don't use it that much because we're usually upright and gravity will do it for us. But the process is in place for peristalsis to help with getting food into our stomach if needed. At the end of the esophagus is a circular muscle called the lower esophageal sphincter. Sphincters are circular muscles that when they are relaxed, they're closed, and when they contract, they open up, generally speaking. And so the lower esophageal sphincter separates the esophagus from the stomach. It's a pretty big deal. If it fails, if it starts to open up, that means the contents of the stomach have the potential to splash up into the esophagus. And the contents of the stomach are ridiculously harsh and acidic and not good for the esophagus. So when that happens, there's consequence. And the consequence is usually pain. And that pain is called either heartburn or acid reflux. And it's all describing failure of that muscle that separates the esophagus and the stomach to keep the contents separate from the esophagus and the stomach. So now we're in the stomach. Once the food's entered, on the other end of the stomach is another sphincter. So I want to at least talk about both ends, right? We talked about the lower esophageal sphincter. On the other end of the stomach is what's called the pyloric sphincter. The pyloric sphincter is also a big deal. It regulates the rate of emptying of the contents of the stomach, right? And so if we just had food drop into our stomach and we didn't have the pyloric sphincter there, food would go straight into the small intestine, and that isn't okay. The stomach has a specific purpose to take the food that you ate and let it get broken down and sort of let it morph into a consistency that the small intestine can handle to then perform its role. If food gets into the small intestine too quickly, digestion goes haywire. And some people have that problem, and that can lead to a lot of digestive issues, which can lead to a lot of other health issues. So the pyloric sphincter controls the rate of gastric emptying. Generally speaking, food stays in our stomach 2 to 4 hours. It depends. Depends on what you ate, depends on how well you chew. And so if you don't chew well, food is forced to stay in our stomach longer. It's not a good thing necessarily. And certain foods, foods high in fat, high in protein will stay in our stomach longer than foods that are high in carbohydrates. So that's the pyloric sphincter. The stomach is a holding tank. Yeah, depending on how big you are, it's somewhere in the neighborhood of 4 to 6 cups of material can be in there. So about 1.5 liters or so of food and drink, liquid. The secretions of the stomach, there's several, hydrochloric acid being one of the main ones. There is an enzyme secreted in the stomach called pepsin. First, it's called actually pepsinogen, and it gets converted to pepsin. This is an enzyme that breaks down proteins. So we have our amylases break down carbs, our lipases break down fats, our pepsin or peptidases will also then break down proteins. We have another secretion called intrinsic factor that helps with vitamin B12 absorption, and there's a couple other factors that are released in the stomach that help with the absorption of some other micronutrients, vitamins, and minerals. In the stomach, the food that we once ate becomes what is called chyme, C-H-Y-M-E. And the chyme is kind of the consistency of oatmeal-ish. So again, visualize whatever you've eaten. It needs to get to that oatmeal consistency before the pyloric sphincter goes, okay, you can leave. You can leave. So the formation of chyme is the end goal of the stomach, of whatever contents enter the stomach. And probably something I definitely shouldn't forget to mention is that lining the stomach is a mucus layer. And this very, very thick mucus layer is why the stomach doesn't digest itself. The contents of the stomach are incredibly acidic. And have these enzymes that break down proteins, which would be the tissue of the inner lining of the stomach itself. Well, this mucus layer protects the stomach itself from it auto-digesting itself. So very important that mucus layer stays intact. And you're probably familiar with conditions that break down the mucus layer. People, when they have ulcers, that's the failure of the mucus layer. Multiple things can cause that. Things like stress, things like too much alcohol. Alcohol will penetrate the lining of the mucus. Alcohol is the only nutrient that we can absorb in the stomach, or at least in a meaningful way. In other words, any food that we've consumed or drunk, right, that's made it into our stomach has not entered our body yet. We're not benefiting from it in a health or energy type of way at all, except for alcohol. Alcohol, 20 to 30% of the alcohol in a drink will enter our bloodstream through the stomach. And again, getting back to ulcers, the fact that alcohol can penetrate that mucus layer over time, alcoholism increases the risk for ulcers quite dramatically. But as a bigger picture comment, nothing's been absorbed into our body yet. Stomach's just a holding tank. We'll make it to where things get absorbed in a minute. A little bit on the anatomy of the stomach. And so the stomach is pretty dynamic. Yeah, it has the same layers like the lumen that we described earlier, but there's also specific cells on the innermost lining of the stomach that are worth discussing, and because they all have a very important role. And so the lining of the stomach is lined with what are called gastric pits. And so the lining of the stomach isn't totally smooth, smoother than the small intestine, but not as smooth as the esophagus. And in these gastric pits, we have different types of cells that sort of line the surface. And so the 4 main cells are the neck cells, and the neck cells, their primary role is to produce all that awesome mucus that is protecting the stomach itself, right? And so as you can imagine, if the neck cells get compromised, they get damaged, whatever happens, trouble's going to follow. There's chief cells, or sometimes they're called peptic cells or zymogenic cells. These are the cells that produce the enzyme pepsin, ultimately pepsinogen, which gets converted to pepsin, that helps break down proteins. And so the chief cells secrete what becomes the enzyme that helps us break down proteins that are in our stomach. And then we have parietal cells. And parietal cells are the cells that release hydrochloric acid and also that intrinsic factor that we talked about that helps absorb by vitamin B12. So they have a very important role as well. The hydrochloric acid, since I mentioned earlier in the mouth how we have some defense mechanisms, the hydrochloric acid, the acidity of HCl, hydrochloric acid, kills most bugs, most bacteria, most things. Not everything. Otherwise, we would never get sick. So certain things pass through the stomach that weren't killed by the hydrochloric acid. But that is another line of defense from invaders, right? From things that might potentially make us sick. So, and then lastly, we have what are called enteroendocrine cells. And these are essentially almost like little glandular cells. They secrete a hormone called gastrin. And gastrin, when it gets secreted, is a hormone, and it stimulates other cells to do their job. And so the neck cells, the chief cells, the parietal cells, generally, they don't just do their thing by themselves. They need to be told to secrete what they secrete. And it is gastrin that gets released by these enteroendocrine cells that does it. So when we eat, when we start thinking about food, right, it's those enteroendocrine cells that get stimulated first, and then the parietal cells and the chief cells. The neck cells are always doing their thing, but the parietal cells and the chief cells will then be told, oh, hey, gastrin's been secreted. We must be getting food soon, so let's start secreting our things. Otherwise, if they're always doing this, we'd have probably too much acid in our stomach, right? And too much pepsin, right? If we don't have protein in the stomach and we have all this protein enzyme in our stomach, it's going to get wasted or even cause damage. So it is gastrin that controls or regulates the release of products from those other cells. Yeah, just a couple pictures of the stomach, not anything to spend much time on, but at least that top graphic, if you can see it, it just shows the orientation of the muscles. And again, the stomach's where a lot of churning happens, right? You figure a couple hours of food to get pushed around, and depending on the orientation of those muscles, the circular ones will squeeze the stomach, the longitudinal ones will push it from top to bottom, that kind of stuff. And ultimately, when the pyloric sphincters opens up, some food will get, or some chyme will get pushed into the small intestine. So finally, I think last slide on the stomach. Yeah, we've mentioned absorption already. Nothing is absorbed other than alcohol. Gastric juice itself, and gastric juice is a sort of a all-encompassing term in terms of what is the content of the stomach. It's not juice, but a collection of all the substances released. They call it gastric juice. So just remember that, yeah, hydrochloric acid is a component, or is one of the things that's released in the stomach. It has multiple roles. It does convert that pepsinogen to pepsin, which is that protein enzyme. It denatures proteins that you consumed initially. So let's say you buy a chicken. You swallow that chicken. The denaturing of proteins is a process where the protein molecule itself is twisted or turned or broken out of its natural conformation. It doesn't break it apart, but it changes the structure of the protein to render its function useless. The protein doesn't serve its original function anymore. It's part of the digestion process. Hydrochloric acid will help release nutrients from organic complexes, and so this is the release of vitamins and minerals that are contained in foods that we consume. It acts as a bactericide, which we already talked about, its antibacterial properties, and also plays a role with iron absorption. So a lot of assistance with allowing us to absorb vitamins and minerals, protecting us from bugs, helping us digest proteins. We've talked about those other things in the gastric juice already, so know them. All right, now we're into our small intestine, right? So we make it through the stomach, pyloric sphincter opens up and does its thing. Now we get into where a lot of the magic happens. Everything in this process is magical, but the small intestine, oh wow. So the small intestine, it can be broken down into three segments: the duodenum, the jejunum, and the ileum. And so the graphic on the right side of the screen here kind of identifies them. The duodenum is that first section. It's about 1 foot long or so. The jejunum is the next section, about 4 feet long. And then the ileum is the final section. It's about 5 feet long. And so if you add those three together, we're looking at, at least 10 foot long, if not longer, organ here. Quite long. Right? It's all curled up in there. When we look at those three sections of the small intestine, they're quite similar overall. If you're just to look at it under a microscope or even on an exam table, but they do have a little slightly different functions. And the duodenum, which is the section of the small intestine that gets chyme recently secreted from the stomach, right, allowed to enter from the pyloric sphincter, its primary role is to bombard that chyme with enzymes, so to let the digestive process get completed. And so the duodenum is where the majority of digestion occurs. There's some absorption there, but most digestion occurs in the duodenum. The jejunum, we're starting to see less digestion, right? Those enzymes that were released in the duodenum, they should have done their thing, and now in the jejunum, we're starting to see a good combination of the completion of digestion and absorption. And then finally, when we get to the final segment of the small intestine, it's all absorption. Like whatever hasn't been absorbed yet better get absorbed now, because guess what's next? It's the colon, and not much is getting absorbed there. So we kind of have three distinct phases associated with those three distinct sort of segments of the small intestine. Some authors disagree on exactly how much digestion absorption is going on between the jejunum and the duodenum. Don't stress. I won't pick on you too much on that. Just know that, yeah, the duodenum gets the chyme first, and the most digestion needs to happen there because the stomach, not tons of digestion is actually going on there. So looking at the anatomy of or discussing the anatomy of the small intestine, the inside of the small intestine is incredible. The walls are folded and dramatically folded, right? It's not like looking through a garden hose where it's completely smooth and you can see through one end through the other. If you took a segment of the small intestine, you would not ever be able to see through it because the folds are that far into the internal aspect of the small intestine. Those folds are covered by things called villi, and those villi are covered by something called microvilli. So the folds have finger-like projections. Those are the villi. And then on those finger-like projections are tiny little finger-like projections, which are the microvilli. The purpose of that is to increase the surface area of the inside of the small intestine. And if you think about it, right, we've consumed some food. We should be designed to absorb all the nutrition from that food, right? To survive as a species, we need to be able to benefit maximally from the food that we consume. Food isn't always in plenty amounts, right? I mean, if we go back a couple hundred years, right, we had groups of individuals on this planet who were starving all the time, right? So as a species, to survive, we need to maximize the potential to benefit from the food we eat and not just poop it all out. So these villi and the microvilli, they increase the surface area of the small intestine to maximize the chance of first food being digested, so broken down into its smallest components, and then ultimately being absorbed into our body so we can benefit from the nutritional value of that food. So some people have mentioned, and again, I don't know if this is an estimate, but surely they do know how to do their math, but they say if you took the small intestine of an average sized person and laid out the complete surface area of that small intestine onto the ground, it would cover an entire football field. That's a lot of surface area for your bagel that you had for breakfast to come in contact with digestive and absorptive cells. So the turnover of the small intestine is pretty quick, meaning that cells in the especially in the lining of the small intestine, live for in the neighborhood of about 21 days, so about 3 weeks. It's a very harsh environment, lots going on there. Therefore, it's a lot of cell turnover. Some pictures of the insides of the small intestine. These aren't pictures, these are drawings, right? But if you would look at the small intestine itself, right, and we take a little cross section, yeah, there's these folds, and they're usually even more dramatic than this. But we have these folds, and then on the folds we have these villi, right? And the villi, they're dense. It's not like they're all spread out. It's like a carpet, right? Each little strand of your carpet is one of those villi. But then if you take a closer look at the villi, we have what are called microvilli. And on those microvilli, which is like a carpet on a carpet, each thread of the carpet, right? Interesting analogy, right? Those are our absorptive cells. And so that's where we want the most broken down components of our carbs, our fats, our proteins, and our vitamins and minerals to come in contact with, because then we have the process of absorption happening. So that's the lining of that's the anatomy of the small intestine in cartoon form. Don't stress about this graphic too much. The enzymes I talk about are the ones that I'll ask you to regurgitate on exams and quizzes and so forth. But this is a decent sort of table or representation of the different digestive enzymes that are part of that digestive process. And so a fair number of enzymes are released in the small intestine. They come mostly from the pancreas. Some are produced by the small intestine itself, and you can see here in the second column it says where the enzymes are created, right? Pancreas down here, small intestine, gastric glands. It says where they're released, and then talks about their pH level, which isn't so important for us in this class. So again, a good graphic or a good table to be familiar with, but in terms of specific enzymes in this class and what I'm going to test you on, they're the ones that I'll mention frequently. Salivary amylase, I would say that's a fair bet. looking at something like a whatever nuclease. I don't talk about those ever, so you probably won't be tested on them. So anyway, a little help. So once the chyme enters the small intestine, gets bombarded with enzymes either made in the small intestine or by the pancreas, released there, our food or chyme gets broken down into its most basic components. Carbohydrates into monosaccharides, either glucose, galactose, or fructose. Proteins into individual amino acids, fats into triglycerides. They get absorbed. What's left over? That's what makes it into the large intestine. And so the large intestine, also referred to as the colon, is sort of the final stage of this process. And so when we leave the small intestine, we have what's called the ileocecal valve. And the ileocecal valve is a sphincter, another one, right? These are important muscles. And it separates the small intestine from the large intestine. This is a pretty big deal. And on the surface, you're like, well, why? Like, things are only going to go one way, not so much, right? And we don't want contents of our colon, of our large intestine, backing up into the small intestine. And more importantly, there's different bacterial flora, so different types of bacteria that exist in our colon in comparison to our small intestine. And the bacteria in our colon need to stay in our colon, as the bacteria in our small intestine do not do well when they get invaded by the other bacteria. And so the balance of bacteria really is regulated by the function of the ileocecal valve. And when it fails, it can cause a lot of problems in people. The balance, and we'll talk about the bugs in our body in a minute. And Sean in The Dark Side of Fat Loss does a good job of talking about it in chapter 8. So anyway, the ileocecal valve, know that it separates the small intestine from the large intestine. Looking at the large intestine, we have sort of three distinct aspects, kind of like the small intestine, right? We have the ascending, the transverse, and the descending parts. And so if we start down here at the cecum, right, the ileocecal valve is right down there. This part would be the ascending, this would be transverse, and this would be descending. We finish with the rectum and the anus. The rectum is where our stool, or your poop, is stored, and then obviously the anus is the pathway for which it is eliminated. Some absorption does occur in the large intestine. We have water absorbed there, sodium and potassium and chloride, so some electrolytes, and also vitamin K. Vitamin K is a vitamin produced by bacteria in the colon. Thankfully, the bacteria makes a vitamin and our body can absorb it there. It'd be a shame if it made the vitamin and then we pooped it out. That would be kind of a waste, right? Water can be absorbed there. So people who suffer from things like constipation, quite often the lack of water or dehydration might contribute to that. As another FYI, and this is, again, some of you probably know Dr. Ping. Dr. Lu, I used to work quite a bit with him in the world of yoga, but we talk a lot about inverted poses, right? So getting inverted and what that does to us physiologically. Well, from a digestive standpoint, inverting is, so doing a headstand, doing a shoulder stand, that kind of thing. Inverted poses or inverted situations is a really effective way to alleviate constipation. Constipation is when movement of the products in your large intestine slows down and you're not going to the bathroom on a regular basis, at least once a day. When we invert ourselves, constipation usually happens in the ascending aspect of the colon, so this segment right here. When we invert ourselves, it allows gravity to help pull contents this way, right? And it's not, we don't need to invert ourselves for 10 hours a day, but even just 2 or 3 5-minute sort of inversions throughout the day help with movement. So you never know. I might ask you that on the exam. So the large intestine, some of its stats, much shorter than the small intestine. It's a little bit bigger in diameter, but definitely shorter in length. No villi or enzymes, very little digestion. Actually, pretty much no digestion occurs here. It is indigestible food stuff that should be arriving. It shouldn't be digestible things, otherwise something went wrong upstream. The most bacteria, at least the highest concentration of bacteria, exists in our colon. And yes, this is where we form feces that are going to be eliminated. So that's sort of digestion from mouth to anus. We discussed in the very beginning that there's 3 accessory organs. So we're going to talk about them now. Those 3 accessory organs are the liver, the pancreas, and the gallbladder. And so this is, again, another nice graphic from a previous edition of Think. They still have something similar to it in your version, but it's just not as good. And so the liver, which is this big one right here, the pancreas, which is this yellow one here, almost looks like a banana slug, and then the gallbladder right here. And so they play a role as accessory organs. So they never come in contact with food, but they absolutely play a critical role in good nutrition and good digestion and absorption. So let's talk about the liver first. The liver, one of its first roles, and it has multiple, is that it produces a product called bile. Bile is like a soap. So soap, if you've ever done the dishes, which I hope you have, when soap comes in contact with fat, you have seen it break the fat up into smaller sort of globules that might be floating on the water. That's exactly what bile does. So the liver produces this bile to help break down fat globules into smaller ones, and that's a digestive process. The liver makes the bile, and it can make it on the fly, and so if we eat a fatty food, liver bile production will increase. But what we actually have, which is fantastic, is that we have a storage place for the bile, and so the bile is stored in the gallbladder. So gallbladder is this holding tank for bile, and so when we eat food, especially food high in fat, the gallbladder will be stimulated or triggered to contract. And when the gallbladder contracts, it will release bile through what's called the common bile duct into the duodenum, right? So into the very beginning of our small intestine. So that's really the process there. The liver makes the bile. It's a fantastic, it's not an enzyme, but it's a fantastic product that breaks larger fat molecules into smaller ones. We hold it in the gallbladder. We eat a fatty food. The gallbladder contracts, and its contraction leads to the secretion of that bile into the upper part of our small intestine, the duodenum, so we can break the fats up. So later in the small intestine, the fats will be broken down enough to hopefully get absorbed. Individuals can live without a gallbladder, and the tricky thing for them is to probably not eat foods too high in fat, or at least high volume of fat in a meal, can pose some challenges for them. There are also medications that help, but the liver will still make the bile. It's just there's no holding point for it. So another function of the liver in the digestive process is what's called the enterohepatic and portal circulation. And so there's a circulatory system that exists between the liver and our digestive tract, more specifically our intestines. And so the small intestine and the liver communicate. And so there's certain products that we consume that get broken down in the small intestine, and by the time they make it to the point where they're absorbed, they are not absorbed in a form that our body can use. And so as an example, fructose. Fructose is abundant in a lot of foods we eat, not just naturally in fruit, but you got sodas and all other kinds of sweets and stuff. So fructose is a sugar, right? And so it can be a part of a complex carbohydrate, it can be part of a simple carbohydrate. But when we consume foods high in fructose, again, whatever food it was part of, it gets broken down smaller and smaller. That fructose, instead of being absorbed and getting dumped into our blood, it goes through this enterohepatic circulation. And so it will transport the fructose from the small intestine to the liver. And what happens in the liver? A little bit of magic, right? The liver will literally change the structure of fructose into glucose and then drop it into our bloodstream. And so this circulatory system now between the liver and the digestive tract becomes really important. It enables us as humans, right, as a living being, to maximize the benefit from the food that we eat. Imagine if we didn't have this process and fructose got dumped into our blood. We can't do anything with it. We can't metabolize fructose. We can't do anything with it. So we'd end up peeing it out or pooping it out. So this enterohepatic circulation is a sort of almost a plan B. It's like, hey, if you consume things that we don't directly have an ability to utilize, the liver will step in, convert it into something that we can utilize, and then put it in the bloodstream where it needs to go to get delivered and utilized from there. So, all right, the pancreas is the next accessory organ. The pancreas is critical, right? And so just like the liver, we can't live without the liver at all. Pancreas is similarly important, for different reasons though. So the pancreas has 3, or I'd say 4 really important secretions. First is digestive enzymes. And from that table a couple slides ago, you saw there's a bunch of enzymes that are secreted by the pancreas, help us break down carbs, fats, and proteins. massive, right? They get secreted into the duodenum, the very beginning of the small intestine, so they have maximum opportunity to come in contact with those macronutrients and break them down into their smallest components for later absorption. The pancreas secretes two hormones, glucagon and insulin. I'm not going to spend a lot of time talking about them in this discussion, but we'll get to them later in the semester. But glucagon is a hormone that helps liberate stored carbohydrates and glucose and drops them back into the bloodstream. So if you're starving and your blood sugar starts to drop and you don't have food to consume to bring blood sugar up, glucagon will get released and help liberate stored carbohydrates and get them back into the blood from their storage form. Insulin does the opposite. Some of you are probably quite familiar with insulin. Insulin helps get sugar or carbohydrates, glucose more specifically, in our bloodstream into the cells, either the fat cells or muscle cells or liver cells. So that's both are released by the pancreas. The third one, that one that's underlined there, is a substance called bicarbonate. And so in the process of digestion, we haven't talked about this yet. The contents of the stomach, we have talked about how harsh they are, and that we need this extra thick mucus lining of the stomach to protect itself, from its own contents. Well, when the pyloric sphincter opens up, those contents haven't changed. It's not like all of a sudden you go through this magic gate and things of, the chyme becomes less acidic and harsh. There is something that helps that process. And what happens is that the pancreas releases something called bicarbonate. Bicarbonate is a powerful biological buffer. And so that acidic nature of chyme, as it enters the small intestine, bicarbonate gets secreted and dumped onto the chyme as it leaves the pyloric sphincter and neutralizes it. And therefore, the chyme that enters the small intestine doesn't burn into the walls of the small intestine, and we don't auto-digest our small intestine. So the bicarbonate that gets released by the pancreas, not talked about a lot, but it is critical. If we didn't release that bicarbonate, we'd be in huge, huge trouble. So pancreas plays a really, really critical role in the digestive process because of all those reasons, but really that chyme and the enzymes especially. So, this graphic is our pancreas right here. We have our gallbladder, right? And like I said before, we can live without the gallbladder. The hepatic ducts will take bile from the liver, it stores in the gallbladder, or it can just bypass that and go straight into the small intestine. Again, we want to keep our gallbladders. But there's a gallbladder right there. And this is the top of the small intestine, right? So this is our pyloric sphincter. So this theoretically going up here would be our stomach. It's obviously not shown. Pyloric sphincter here. So food entering from the stomach enters in through this direction. And we have here our pancreatic duct that breaks off into our pancreatic duct right here. And then also our duodenal papilla, which is also another duct or another exit for the enzymes, and in this case, the bile will leave through those different ducts. So, communication between them, these accessory organs, how do they get in contact with the food? Well, their secretions will enter through these ducts into the duodenum to mix with the contents leaving the stomach. Good stuff. All right, we're going to stop here. We'll pick up part 2 with the next lecture.